Welcome back to the Deep Dive. Today, we are taking what I think is one of the great conceptual leaps in the history of biology.
Whoa, for sure.
We are finally moving past the theoretical abstract concept of the gene Mendel's invisible hereditary factors
Right.
and diving directly into the physical mechanics of it all.
That's exactly right. For decades, the rules of segregation and independent assortment were just, mathematical concepts. Our mission today is to solidify that connection.
Okay.
We're drawing from core texts on the chromosomal basis of inheritance, and we're going to prove that those abstract factors reside on and are carried by concrete physical structures.
The chromosomes.
The chromosomes.
So we're essentially synthesizing the critical breakthroughs of the early 20th century. We'll unpack 5 key concepts, starting with the foundation, moving through sex-linked traits, gene mapping,
And then finishing up with some genuinely strange exceptions to the standard Mendelian rules.
It's a deep dive into how the tiny, precise dance of chromosomes during meiosis really governs everything we are.
The breakthrough that started it all is known as the chromosome theory of inheritance.
Right.
This theory came together around 1902. You have 2 researchers, Walter Sutton and Theodore Boveri, and they're working independently.
And what did they find?
They realized that the movements of chromosomes during meiosis perfectly mirrored the rules Mendel had established decades earlier.
So what were those key parallels that were just, too compelling to ignore?
Well, think back to meiosis the 6th. Mendel proposed that hereditary factors come in pairs.
And so do chromosomes.
Exactly. And Mendel's first law, the law of segregation, says the 2 alleles for a trait separate into different gametes. This maps perfectly to how homologous chromosomes separate during anaphase 3rd of meiosis.
That makes so much sense. The 2 members of homologous pair pull apart, and they carry their specific alleles with them.
Precisely. Then you have Mendel's second law, independent assortment.
That alleles for different traits sort independently.
Right, and this is explained by the random independent alignment of those homologous chromosome pairs at the metaphase plate.
So whether the chromosome with a red allele goes left or right is totally independent of where the one with the short allele goes.
You got it. The conclusion was unavoidable. Mendelian genes must have specific physical locations or loci along the length of the chromosomes.
Theory is one thing, and experimental proof is another. To prove this link, they needed the perfect experimental subject.
And that's where Thomas Hunt Morgan and the fruit fly, Drosophila melanogaster, enter the scene.
Why the fruit fly?
Morgan's choice was just genius. These tiny flies are incredibly easy to raise. They breed prolifically.
So you get tons of data.
Hundreds of offspring and a very rapid generation time. And crucially for his work, they have only 4 pairs of chromosomes.
Only 4, okay.
3 pairs of autosomes and 1 pair of sex chromosomes, XX for female, XY for male.
Morgan was looking for natural variations, right? What he called mutant phenotypes against the standard wild type.
That's it. The wild type is just the trait you see most commonly in nature. After months of searching, he finally found a single male fly with white eyes instead of the typical red.
Just one.
One fly. He designated the wild type red allele as W, plus that plus sign just means wild type, and the recessive mutant white allele as W.
So he performs his initial cross. A wild-type red-eyed female mated with his one mutant white-eyed male.
In the F1 generation, they all had red eyes.
Okay, so red is dominant. Standard stuff.
Standard Mendelian fare. He then interbred the F1 generation to get the F2s, expecting that standard 3.1 ratio of red to white.
And did he get it?
He did get the 3.1 ratio overall, and here's where the story just diverges dramatically when you look closely.
What?
All of the white-eyed flies in that F2 generation were male.
Wait a minute. All of them?
Every single one.
Now, hold on. If this gene were on an autosome, a non-sex chromosome, you'd expect the white eyes to just pop up randomly in both sexes, right? In equal measure.
You would.
So why would it be exclusively linked to the male flies?
That observation, that was the definitive proof. It meant the gene for eye color wasn't on an autosome. It was located specifically on the X chromosome.
Ah, and because the male fly only has one X chromosome.
And a Y that lacks the eye color gene, he is completely dependent on that single X copy to determine his eye color.
So if the male inherited a single recessive W allele on his X, he would automatically have white eyes because the Y chromosome can't mask it.
Exactly. That one experiment provided the first solid physical evidence associating a specific gene with a specific chromosome. It was the foundation of sex-linked inheritance.
So moving from fruit flies to humans, These unique patterns must be magnified, right?
Oh, absolutely. The human chromosomal basis of sex is the XY system, so females are X, males are XY. The X chromosome is large, carries over 1,000 genes, while the Y is just significantly smaller and pretty gene sparse.
But I remember learning that even though the Y is tiny, it contains the critical switch that determines sex.
It does. The critical factor is the SRY gene, the sex-determining region of Y.
S or Y.
Its product is required to trigger the development of testes. If it's absent, the gonads just automatically default to developing as ovaries.
Fascinating.
Now, a sex-linked gene is any gene located on either the X or the Y chromosome.
But we mostly talk about X-linked genes because there's something like 1,100 of them. U-linked genes are minimal, around 78?
That's right, and they're pretty straightforward. They're just passed virtually intact from father to all his sons.
Right.
But X-linked inheritance shows these unique, often complex patterns because of the dosage imbalance. Males only have one X chromosome.
Meaning they're homozygous for X-linked genes. Let's pause on that term, homozygous. What is the consequence of having that single copy?
It means that for X-linked traits, there's no second chance. If a male inherits a single recessive allele for an X-linked disorder from his mother, he has no corresponding dominant allele on a second X to mask it. He will express the trait.
And this is why X-linked recessive disorders like red-green color blindness or something like Duchenne muscular dystrophy.
The devastating muscle wasting condition.
Right, which affects about 1 in 5,000 males, why they're so much more prevalent in the male population.
Exactly. A carrier mother has a 50% chance of passing it to her son, while a father who has the trait will pass the allele to all his daughters, but none of his sons.
But wait, this raises a problem for females. If they have 2 X chromosomes and males have 1, shouldn't females produce twice the amount of X-linked proteins?
That's a great question. If that were the case, females would be biologically different and could, suffer from a protein overdose.
So how does biology solve this genetic dosage imbalance?
Through a really elegant mechanism called X inactivation. Early in female embryonic development, one of the 2 X chromosomes in each cell is randomly and independently selected, condensed, and largely inactivated.
And this condensed structure becomes a visible, compact object called a Barr body.
The Barr body, yes.
So every somatic cell in a female only has 1 functional X chromosome, making the dosage equivalent to males.
Exactly. And because the inactivation is random and independent in each cell, females are essentially a mosaic. Some cell patches express the alleles from the paternal X, and other patches express the alleles from the maternal X.
And the classic visible example of this is the tortoiseshell cat.
It is the perfect illustration. The gene controlling black versus orange fur color is X-linked.
Okay.
If the cat is heterozygous, so it's carrying both the black allele and the orange allele, the random inactivation means that wherever the cell lineage keeps the X with the orange allele active, the cat has orange fur.
And where the other X is active, the fur is black.
You get that characteristic patchy coloration, which is why tortoiseshell cats are almost always female.
Okay, so we've placed genes on chromosomes. Now we have to deal with the fact that a single chromosome carries hundreds or thousands of genes.
Right.
Which brings us to linked genes. Genes located close together on the same chromosome that tend to be inherited together as a unit.
And Morgan decided to test this linkage hypothesis using a second experiment, this time with 2 traits that were not sex-linked: body color and wing size.
Okay, so B plus for gray, B for black, and then VG plus for normal wings, VG for vestigial or small wings.
Right. And both genes were located on the same autosome. So he performed a test cross. He took F1 dihybrid females who were heterozygous for both traits and mixed them with homozygous recessive males.
Black body, vestigial wings.
That's the one. Now, if these 2 genes sorted independently, if they were on separate chromosomes, he would have expected a 1.1.1.1 ratio in the offspring.
So 50% parental types, 50% new combinations or recombinant types.
But that's not what happened at all.
What did he see?
The offspring were overwhelmingly parental types. Gray normal and black vestigial offspring made up about 83% of the total.
83%, that's a massive skew.
It proved that the 2 genes were indeed physically linked on the same chromosome.
But wait, if they're physically linked, why isn't the link absolute? Why did he see any recombinants at all?
Yeah.
If linkage means they stick together, why were there still about 17% of offspring with new combinations like gray body vestigial wings?
That 17% was the second huge conceptual leap. It meant the linkage wasn't absolute, it could be broken. And the mechanism breaking the linkage was crossing over.
This is the physical exchange of corresponding segments between non-sister chromatids that happens during prophase of meiosis.
Exactly. The chromatids physically break and rejoin, shuffling the alleles and creating chromosomes with new combinations of alleles, the recombinant chromosomes.
Which explains the non-parental types.
Precisely. And it was Morgan's brilliant student, Alfred Sturtevant, who had the revolutionary insight. He hypothesized that the recombination frequency, that 17% for body color and wing size, was directly proportional to the physical distance between the two genes on the chromosome.
That's fascinating. So the farther apart the two genes are, the more room there is for a crossover event to happen between them, which leads to a higher recombination frequency.
That's the core idea. This allowed him to create a linkage map, which is an ordered list of genetic loci along a chromosome, all based on recombination data.
And he defined a map unit as equal to a 1% recombination frequency.
So if body color and wing size recombine 17% of the time, they are 17 map units apart on that chromosome.
So this is how geneticists can map the relative positions of genes without even looking at the physical chromosome. It's just statistics.
It's pure statistical mapping. But it's also crucial to remember that if two genes are located very far apart on the same chromosome, say 50 map units or more, they have a 50% chance of recombination.
In which case they're considered genetically unlinked.
Right, because 50% recombination is indistinguishable from the result you'd get if the genes were on entirely separate chromosomes assorting independently.
So while all this usually works, errors in cell division can lead to large-scale chromosomal changes.
And those changes can result in severe disorders. The primary error mechanism is non-disjunction.
Non-disjunction.
It's the failure of chromosomes to separate properly. This can happen in meiosis the first, where homologous chromosomes fail to separate, or in meiosis the second, where the sister chromatids fail to separate.
And when a gamete with an abnormal number of chromosomes fuses with a normal gamete, the resulting zygote gets a condition called aneuploidy.
Right, an abnormal number of a particular chromosome. The two main conditions are trisomy, which is 2n+1, so one extra copy of a chromosome.
And monosomy.
2n1, where one copy is missing. Monosomy is almost always lethal in humans.
I see.
And then you have polyploidy, where an organism has more than two complete sets of chromosomes. Think triploidy, 3n, or tetraploidy, 4n.
And plants, like bananas, tolerate that much better than animals.
Do. Now, beyond number issues, chromosomes can also suffer structural changes from breakage and rejoining errors.
Okay, what kind of errors are we talking about?
We define 4 types. First, deletion, where a fragment is just lost entirely. This is usually lethal if the fragment is large.
Just a second.
Duplication, where the fragment attaches as an extra segment somewhere else. Third is inversion, where the fragment reattaches to the same chromosome but backwards.
And the fourth is translocation, where a fragment joins a non-homologous chromosome.
That's right. And reciprocal translocations, where two non-homologous chromosomes just swap segments, are the most common type.
In terms of human conditions, the most well-known example of aneuploidy is Down syndrome.
Down syndrome affects about 1 in 830 births and is almost always caused by trisomy 21, an extra copy of chromosome 21. We know the incidence of this non-disjunction event increases dramatically with the age of the mother.
Interestingly, he said sex chromosome aneuploidies, having too many or too few X or Y chromosomes, are tolerated far better.
Much better, yeah. That's due to the small gene count on the Y chromosome and that efficient X inactivation mechanism we talked about.
So you get conditions like Klinefelter syndrome, XXY,
which results in males who may be sterile and face some learning difficulties, and Turner syndrome, XAIR, which results in females who are sterile but often have typical intelligence. It's actually the only known viable human monosomy.
And finally, there's a critical example of a structural change leading to cancer. Chronic myelogenous leukemia, or CML.
CML results not from a meiotic error, but from a reciprocal translocation that happens during mitosis in white blood cell precursors.
So it's acquired, not inherited.
Exactly. Chromosome 9 and chromosome 22 swap segments, resulting in a distinct, shortened structure called the Philadelphia chromosome.
The Philadelphia chromosome.
This exchange fuses two genes together, creating a new, highly active hybrid gene that drives uncontrolled white blood cell division. It's a direct link between chromosomal structure and disease.
We've established the rules, but biology always has exceptions. We have to address 2 ways inheritance can just completely defy Mendelian expectations.
Right, where the sex of the parent does matter, sometimes even for genes on autosomes.
The first exception is genomic imprinting.
This is where the phenotype expressed by an allele depends entirely on which parent contributed that allele. Which is confusing because Mendelian genetics tells us an allele is an allele no matter where it came from.
So how does the cell remember which parent a gene came from?
It's an epigenetic mechanism. During gamete formation, either the maternal or the paternal allele of a specific gene is effectively silenced or marked for expression.
How?
This often involves the addition of methyl groups to the DNA nucleotides, a process called methylation that essentially just turns off the gene.
Which means even if an offspring is heterozygous, only one allele is truly available for expression, depending on the sex of the parent it came from.
Precisely. A famous example is the mouse EGIV2 gene, which codes for insulin-like growth factor 2.
Which is critical for prenatal growth.
Right. For this gene, only the paternal allele is expressed. The maternal allele is always imprinted and silenced.
Wow.
So if a mouse inherits a mutant dwarf allele from its father, it's a dwarf. But if it inherits the exact same mutant allele from its mother, it is normal-sized.
Because the maternal allele would have been silenced anyway, and the paternal copy is the active wild-type one.
It's absolute genetic memory, and then the functional imprint is completely erased and reset based on the sex of that individual before they form their own gametes.
Incredible. What's the second exception?
The second exception takes us completely outside the nucleus. We're talking about extranuclear, or cytoplasmic, genes. These are the genes found in the small circular DNA of mitochondria and, in plants, chloroplasts.
So since these organelles have their own DNA, their inheritance patterns must be different.
They are. These genes display strictly maternal inheritance.
Why maternal?
Because the egg contributes the vast majority of the cytoplasm and all the organelles, including the mitochondria, to the zygote. The sperm contributes almost nothing but the nucleus.
So if a plant has a mutation in its plastid DNA that causes variegated or patchy leaves, that coloration will always be inherited from the mother plant.
Correct, regardless of the father's genotype. And in humans, mitochondrial disorders, which might affect vision or cause muscular weakness, are almost always traceable through the maternal line.
And this all comes down to energy, right?
Yeah.
ATP production.
It does. The cell's energy supply is compromised. This is why you hear about research into technologies to replace faulty mitochondria in an egg, resulting in what the media sometimes calls three-parent babies, to prevent transmission of these severe maternal disorders.
What an immense set of findings we've synthesized today. I mean, we confirmed that the abstract rules of Mendel are physically rooted in the movement of chromosomes.
And we learned that X-linked genes require their own unique rules, often leading to dosage compensation through the Barr body and that mosaic nature of females.
We saw how Morgan's team, just by tracking linkage and recombination frequencies, developed the concept of crossing over and the revolutionary technique of genetic mapping.
It allowed us to determine the relative distance between genes. It's just incredible.
And then we covered the devastating consequences of errors, from the meiotic mistakes leading to aneuploidies like Down syndrome, to mitotic translocations that can cause cancers like CML.
And finally, we saw how intricate biology can be with those exceptions, like genomic imprinting and maternal inheritance.
It's fascinating.
To close, let's go back to that EGF2 gene and genomic imprinting, which favors the paternal allele for maximizing offspring size.
Right.
This strongly suggests a silent evolutionary conflict. Some scientists propose that fathers evolve strategies to maximize the size and resource capture of their offspring, potentially at the expense of the mother.
Who has to manage her own limited resources.
And the physical strain of delivery. So it really makes you wonder, what does this deep underlying genetic tension, which manifests through these complex epigenetic mechanisms, suggest about the fundamental conflict between maternal and paternal genetic strategies throughout development?
That is a complex interplay worth mulling over.
It is.
Thank you for joining us for this crucial deep dive into the physical basis of heredity. We hope this has been your shortcut to being well-informed.